Is the audio okay now? I suppose there was an audio issue. I believe it is sorted, but at least till then everyone saw what I was writing, I suppose. Yeah. Okay. So today's class, like I was saying, today's class is the primarily for INI-CET, but going forward, all the other classes are going to be for INI plus FMG students. So I've given you the details. 30th March को एक और class है, and April 3rd to April 6th is going to be a TND for Path and Micro. Two classes of Path, two classes of Micro that are going to be conducted. Right? I hope that's okay with everyone. So chalo, let us begin. It's a very short session. Quickly, you have to. Acha, there's a volume issue. Just a second. I believe that's the maximum volume, guys, that this can, in a way, take up. So I hope it's manageable, or is it like really low on the sound? Volume is okay? Right, chalo. As long as it's manageable, because it's, as I said, it's not a very lengthy session. So you're going to answer some quick questions, and I'm going to make you answer this table. ये table पहले करना है, the CD markers वाला table. These two you will fill up. After that, flow cytometry is going to be a cakewalk. So पहले tell me the main things. You can see this flow cytometry happening over here. Quickly you're going to tell me कि flow cytometry में जो laser light use होता है, what is the light, what is the wavelength? Question number 1. Previous year INI-CET, what color laser light is used? It is a blue color laser light which has a wavelength of 488 nanometers. The first question that has come in your paper, right? Coming to the next question, they will ask you, what are all these colors? You don't have to know the names of blue, green, yellow, सब कुछ नहीं पता करना. You just have to know what is the green color dye, what is the fluorescent dye that we use. That is FITC. Please note, FITC is the green color dye which they ask you, because they really like to ask you fluorescence के ऊपर questions ना. In microbiology also they like to ask you fluorescent microscope. So same way over here, they love to ask you this. This is fluorescent isothiocyanate. Just go through the name once, because just in case it comes as a full form, you should be able to answer that FITC is going to be fluorescent isothiocyanate. So the green color dye is also done. Perfect. Now coming to the third question which will come in this table. They are saying, can you see this GIF, guys? How are the cells falling? The cells are only falling one by one. Cells are not falling ek saath, 1,000 cells falling. No. What all have you put inside it? You'll ask me what all sample. Anything fluid can be added. That could be blood, that could be bone marrow aspirate, that could be FNSE, that could be anything fluid can go inside this machine. That is why you call it flow cytometry. And it's going to fall one by one. So why is it falling one by one? Very good. Single sheath it's going to fall. That focusing is known as hydrodynamic focusing. So it sounds like a very thoda sa complicated name, but it's nothing complicated. What is hydrodynamic focusing? Focusing means one cell, one by one, the cell is gonna fall over the light. And how did that happen? Obviously, if you have 1,000 cells, they should have fallen together. Why did they fall one by one? Because you have used a fluid called sheath fluid. Bus. That is where the story ends. You have used one special wala fluid. Like kya kara? You took your blood, you added sheath fluid, and now the blood is gonna fall one by one sample karke, one by one cell will fall. This is also done. Now what is going to be question 4 and 5? Then the story is only khatam. When this light is coming, a part of the light is going forward, a part of the light is going sideward. Everyone can see that. This is the most beautiful GIF which explains the entire machine in one go. So what is forward scatter? What is side scatter? Very good. So now you need to tell me the question number 4, guys. Question number 4 is FSC, forward scatter. Forward scatter is a measure of size. This is the same question that I taught all of you a few days back over Instagram also. Forward scatter is a measure of size. Means if the chota size is there, size of the cell is small, the forward scatter will be chota sa. If the size of the cell is big, then the forward scatter is going to be more. So simple. This is a previous year INI question. Forward scatter is going to be a measure of size. Whereas talking about question number 5, the last machine wala question, what is side scatter dependent on? So how is this going all sideways? Side scatter is dependent on how complex your cell is. Okay, if you say that ma'am, my cell has a lot of granules, a neutrophil hai, या फिर even more basophil hai, lots of granules it has, I'll say it's very complex. It's a very complex cell, so the side scatter will be more. So as much as the complexity, or in simple words, the granularity, more is going to be the site scatter. So that is going to be the last point. Quickly guys, 5 questions and you're done with the machine. Question number 1, quick recap. The light used, blue color light used hua hai, which 488 nanometers. The fluorescent dye used is FITC. The single cell by cell is known as hydrodynamic focusing. Forward scatter is a measure of the size and side scatter is a measure of the complexity. 5 questions are done. Before I make you go on to the charts, you will have to now do the most deadly table that is there, because without doing this table, no one can do the direct analysis. Doing analysis is very childish. Graphs, though anyone can read now, but you need to know what to read. So I know this is a table all of you are very scared of, but you will have to. You cannot escape it. Let's try to make it a little easy. Chalo, what is the thing that is gonna be for blast? So now first ratta, this doesn't have a mnemonic. Everyone ratta lagao. This one column, everyone's gonna mug it up with me. And what is that known as? Anything that has TDT, HLA-DR and CD34. These three things you will have to first lagao ratta so that your life becomes a little easy. TDT, HLA-DR, CD34. Any of these three things written, the examiner is trying to tell me about a blast. So my next job will be blast to hai. I will have to find out, is it a TALL wala blast, BALL wala blast or AML wala blast. So now my job is which kind of blast is it. Everyone rat liya TDT, HLA-DR and CD34. Now we come to something known as LCA. Guys, LCA word only tells me it is a leukocyte common antigen. Common basically means every leukocyte is gonna have it, and that is nothing but CD45. This means whether it is a leukocyte is what? White blood cell, na? So whether it is a neutrophil or a lymphocyte or any white blood cell, every white blood cell will have CD45. Okay. What are the markers of T cells? T cells को कैसे याद करेंगे? 1 to 8, 28. That is how you learn it. It sounds like a rhyme. 1 to 8 and 28. That is going to be the T cells. However, for every lineage, I will tell you one hero. That hero you have to learn. The pan T cell marker is what, guys? Pan T cell marker is CD3. Very good. So repeating 1, 2, 8 and 28 and pan T cell is going to be CD3. Now coming to B cell. B cell में क्या करना है? Double. That is how you will learn it. Double means first you start from CD10, then 10 का double 20, 20 का double 40, 40 का double 80. That is how you will go. So 10, then 10 का double 20. So 20s में you will have all the 19 से लेके 19, 20, 21, 22, 23. All of these are going to be there. Then 20s के बाद double is 40. So the next marker is going to be CD 40 and 40 ka double is 80, but I don't reach 80. I call it 79A and 79B. Instead of reaching 80, I have split that 79 into 2. So repeating once again, you have 10, then 20s, then 40 and then 79. But yes, the next question is which is the hero of this story? Which is the pan B cell marker? CD19. Guys, after some time you will realize no one will ask you this story. Only the heroes will be asked. So heroes पे extra emphasis. The pan T cell marker is CD3 and the pan B cell marker is CD19. Okay, let's go to NK cells, natural killer cells. Come on, you know this. Natural killer, they are killing. They have like wo typical gym wala body they have. Bodybuilder, natural killer cell. So gym wala body matlab that the biceps are gonna be 16 inches and that meme that came out of 56-inch chest. So natural killer cell is a total bodybuilder, that is 16-inch bicep, 56-inch chest. So 16 and 56. And last INI-CET had a new question. Guys, what is someone who has a 16-inch bicep and a 56-inch chest, very bulky person, uska weight to zyada hi hoga. So that is gonna be CD94. That was the recent most question of INI for the NK cell. So 16, 56 and 94 for the bodybuilder that we have. That is the strong person, natural killer cell. Now these three, which are your deadly dreaded ones, okay? Myeloid cell. Myeloid cell के लिए तो simple, ma'am. This is the easiest, my favorite, MPO. Myeloid cell, myeloperoxidase. I wish such questions come in the paper, but they will not. They will ask you some CD markers. 1333. Myeloid के लिए simple है. 1333, CD13 and 33. But they will always ask you the hero. Guys, who's the hero over here? The hero over here is myeloperoxidase. Myeloid ka hero is gonna be myeloperoxidase. So these three heroes will help you solve all your graphs. T ka CD3, B ka 19 and myeloid ka MPO. Okay guys, what is this? This is very interesting. Monocytic lineage. How do you learn monocytic? Very good. I've got some amazing answers. So mono we had learned earlier also, before your NEET also I made you remember this. Mono matlab single, forever single. So what did we say? CD11. 1 matlab single, right? 14th February ko bhi single, that is CD14, and for single till he dies. So 64 ki age may be single. That is what we had studied. Dr. Aditi, the first line I told you today was okay, the flow cytometry class is not very important for FMG. This is more for INI-CET, right? Okay, so coming back. So क्या पढ़ा? Mono. Mono मतलब single. 1 is single. 14th February को भी single and single till he dies. So 64 is also single, right? Finally, mega. Mega karyocytic. The name is only sounding mega. So this we had learnt by the mega gun of PUBG. Do you remember? I told you कि PUBG में एक gun होता है which is 416, right? M416 एक gun होता है PUBG में. So you have to think of mega PUBG, right? So remember that mega gun M416. ऐसे पढ़ो 41, उल्टा पढ़ो 61. So mega gun is gonna be 41 and 61. बीच में 41 का brother 42 also comes. ठीक है? So mega gun is what you will remember, the 416 gun of PUBG. 4161. So 4161 and बीच में उसका brother 42. So at least one part of the table is sorted ना. ये वाला part हो गया sort. Now I'm left with the other part before I start with the graphs. You have to now come down to the different leukemia lymphomas. This will come to you as a graph for sure. So tell me one thing. There is CLL, SLL, mantle cell lymphoma, marginal zone lymphoma. So these are the three that they always ask you in the flow cytometry graph. So repeating CLL, SLL, mantle cell, marginal zone. So dekho, the markers that we are going to study, I'll make a list over here. There is CD5, there is CD23 and CD200. Three markers. This is also an INI-CET question. We have studied that for CLL, SLL, सब कुछ positive. सब कुछ positive means 5, 23, 200, everything is gonna be positive. Margin, so CLL sounds like S na, CLL, SLL, सब कुछ positive. Margin को ignore करो, ignore the margin. This means everything is gonna be negative. Ignore the margin, everything is negative. And mantle cell lymphoma only because the number of alphabets that it has are roughly 5, 6 na. तो सिर्फ 5 पॉजिटिव रखना, बाकी सब नेगेटिव. दैट स्टोरी इज ऑल्सो डन. रिपीटिंग, CLL, SLL सब कुछ पॉजिटिव, एवरीथिंग पॉजिटिव. मार्जिन को इग्नोर करो, सब कुछ नेगेटिव हो जाएगा. And mantle 5, 6 alphabets, only 5 positive, rest everything negative. So this part is also done. Okay, so it should give you confidence. Almost coming towards the end. Erythroid cells, what about RBCs? RBCs have something called transferrin receptor. Agreed? RBCs have transferrin receptor. So now the way you write transferrin, T से सब कुछ आ जाएगा. T के अंदर what have I fit? 71. So the way you write T, what did I do? The way you write T, I have put a 7 and 1 in it. So RBCs के लिए CD71, that is transferrin receptor. Sorted. PNH, come on, this you know, baby question है, jaldi quickly tell me, what is PNH? Perfect. There is absence of something. CD55 and CD59 kindergarten question, no problem. CD55 and 59 are negative. तो ये तो जल्दी से हो गया. Good. Now coming to this Glanzmann thrombasthenia. You will say, ma'am, it is a platelet disorder. Why are you studying it under flow cytometry? पहले guys tell me what is Glanzmann thrombasthenia. You will say, अरे this was the GP2B3A defect. This was GP2B3A defect. So now you will say that why are we doing GP2B3A defect under CD markers? Because what is GP2B and 3A? GP2B and 3A, platelet का सोचो, अभी I told you ना platelet का. Let me go back. प्लेटलेट का मार्कर्स क्या था? प्लेटलेट मतलब मेगा कैरियोसाइट का वो M416 वाला गन 4161. व्हाट इज 4161? नथिंग बट GP2B3A. 41 and 61 is nothing but GP2B3A and that will be absent, matlab there is Glanzmann thrombasthenia. Now coming to plasma cells and multiple myeloma. Difference क्या है? I want you to know the difference between a normal plasma cell and a cancer वाला plasma cell. Normal अलग है, cancer अलग है. So what are you gonna do? You're gonna make a tiny little table over here and I'm gonna tell you a story. Last story and then you are good to go with the graphs. Then I'm gonna tell you a story of a good child and a bad child. A good child is someone who is gonna take care of his parents and a bad child is someone who's gonna leave his parents. तो पहले तो tell me plasma cell के parents कौन है? You will go back to the previous slide. You will say, ma'am, parents. The one parent of everyone is gonna be LCA. Leukocyte common antigen is present everywhere. So one parent definitely has to be LCA. So we will say कि normal plasma cell CD45 positive. Plasma cell comes from what? Plasma cell comes from CD19, मतलब B cell ना, plasma cell actually B cell से आता है, तो B cell उसका parent है, तो CD19 has to be there. So basically 45 positive, 19 positive, ये parental markers हैं. Then after every parental marker you have to have your own identity. So what is the own identity? 38 and 138. CD38 and 138, both of them are gonna be positive. That is apna identity that this cell has. But now when we go to cancer, cancer is a bad child. Cancer will not take care of his parents. तो सबसे पहले it will say CD45 को करो negative और 19 को करो negative. Forget the parents. Only focus on your own self. It will only keep 38 and 138 and it will forget the parents. So simple. A normal plasma cell is a good child. It will take care of parents also. It will take care of itself also. Whereas a malignant plasma cell is a bad child. He has forgotten his parents. He is only focusing on himself. Everyone's understood this story, because now all these stories are going to help you in your flow cytometry graphs. Before I start, how many types of flow cytometry graphs do you have? That is actually question number 6 from the machine. The 5 questions we had done earlier, na? Now I'm going to come up with the sixth question over here. What graph is this? So when you will see that cells are present like this, you will see all flow cytometry ka graphs na have these dots. So this is referred to as a dot plot analysis. This came 2 sessions ago. What is this? This is a dot plot analysis. ठीक है? तो अब इसमें क्या होगा? There will be something written on x-axis like CD3 लिखा है, and something written on y-axis. For example, CD34. So some markers will be written. Whenever the cells are falling here, means you can trace it down. This part high expression, this part high expression. So this means this is gonna be double positive quadrant. DP quadrant here. X is also positive, Y is also positive. If the cells are falling over here, chota chota low expression, this is the double negative quadrant. Everything is negative. बस ये जल्दी से एक बार revise कर लो, then I'm taking you to questions. Everything is over here is double positive, everything over here is double negative. The cells which are over here, they are closer to the x-axis na, so only x-axis is positive, y-axis nahi hoga, only x is positive. And the cells which are over here and closer to the y-axis, only y-axis is positive, not the x-axis. So everyone's understood the difference. This quadrant is DP double positive, this quadrant is DN double negative, this one is only x positive, this one is only y positive. ठीक है? अब now this was one method. What is this? This is the dot plot analysis. Is that the only graph? No. The second graph that you have is this kind of a graph where you will not get that dot dot dot, you will get something else. And so many of you waiting for that CD40 celebrity question. वो आएगा. Don't worry. So this is the second type of graph. That graph is known as a histogram. That is different. So this reminds you of your PSM graphs. जैसे peak में show करता है. So what is this? Can you tell me for what have I done it? Although अगर ध्यान से देखो तो लिखा है on the picture, but what is this eosin 5 malamide? ये किसका testing है? Eosin 5 malamide is a confirmatory test for hereditary spherocytosis. So you will say, okay, flow cytometry helps me in this also. It helps me in RBC, platelet, WBC, सब problems में help करता है. So eosin 5 malamide, as you can see, in a normal person, the color will be too much, and in a hereditary spherocytosis person, the eosin 5 malamide color, why am I saying? Because eosin 5 malamide is a dye. So in a normal RBC, the color will be more. In a defective RBC, the color will be less. When you're getting such peaks, that is going to be histogram. So last question, guys, tell me what are the two types of graphs that you get in flow cytometry? One is your dot dot wala dot plot analysis, and the other is going to be a histogram. Sorted? Now finally I'm going to take you to questions, but pehle you will have to tell me theoretical questions. Okay, then I'll take you to the graphs. Acha, theory question mein one of them I will solve for you. Before I start, instead of getting you lengthy questions, I've got you the calm ki baat. That is, this is what you will see in your question, and this is what you're gonna mark over here. Okay? So now tell me, over here, I can see a lot of markers written. Before you start, you have to follow a rule, okay? First, you will have to pick up blast hai ya nahi hai, and then you will have to find out are the heroes of the story there or not there. Heroes matlab B cell hero, T cell hero, and myeloid hero. Jaldi se quickly tell me what are the myeloid heroes that, or what are the heroes that you have. तो पहले B cell का hero CD19, T cell का hero CD3, and myeloid का hero MPO. पहले तो heroes देखना, you have to find the hero in the movie. And blast क्या है? Do you remember? CD34, HLA-DR and TDT. मतलब this तो should be embedded in your brain as it is. कि blast ढूंढो, अगर blast है तो heroes ढूंढो. So tell me something. This person is CD34 and HLA-DR positive. CD34 and HLA-DR positive. So is this certain that blast पक्का है? Okay ma'am, blast तो है. Now when I started reading 1333, ये तो myeloid का है, but I don't give the diagnosis till I don't find the hero. Yes, I can find the hero over here. Myeloperoxidase is the hero of the myeloid lineage. So what kind of blasts are these? Myeloid blasts. What is the diagnosis? Very good. This is a case of AML. Let's practice one more. We'll get more confidence. Okay, look at this one. CD34 positive. क्या interpretation है? CD34 HLA-DR TDT. मैम blast तो पक्का है. Blast है. And 19 and 10, these are B cell का markers. But hero ढूंढो. Hero है? Yes, hero is there. 19 is the B cell वाला blast. So what is gonna be the answer over here? This is a case of BALL. Okay, let's try and practice some more questions over here, which is gonna be quite an interesting. So simple है. Blast ढूंढा, hero ढूंढा, answer हो गया. But what happens when you have this kind of a scenario? Let's go in the same manner. CD34 positive, HLA-DR positive, TDT, सारे ही blast वाले markers है. Ma'am, this is पक्का blast. कौन सा वाला leukemia? अब 19 is the hero of which lineage? B. I thought अच्छा BALL होगा. But then myeloperoxidase is also there, and that is the hero of the myeloid lineage. Ab how can I have two heroes? हो सकता है ना? This is a blockbuster movie. It has two heroes in it. Now this means इसमें BALL का भी features हैं, and this में AML का भी feature है. So there can be some cases which are known as the biphenotypic leukemia. This is what is a typical AIMS वाला question where they'll give you a blockbuster movie. Both the heroes are there. तो simple. अगर से एक hero होता, one hero is there, one leukemia. Now over here, because you have two heroes in the story, you're dealing with a biphenotypic leukemia. Everyone sorted with this? Because now this was your theory wala question. Quick thumbs up. Everyone's okay? Theory wala questions ho gaya. Can we go on to the graph wala questions now? That is the last part of this class left. So let's begin with this. Let's try and solve it. What do I do in the exam? Ab graph wala stories mein na hero is not important. Whatever information has been given to me, give your answer on the basis of that. It's the easiest thing to do in the paper, guys. So look at this. क्या क्या लिखा है CD10 and your mind will take you towards the lineage automatically. CD10 तो B cell का marker था, and TDT मतलब blast का marker है. So they are trying to tell me some blast and some B cell marker, and they will either put an arrow that which is the quadrant you have to analyze. तो over here arrow पड़ा है, नहीं हुआ arrow. You will say ma'am arrow नहीं था. For example, there was no arrow. Which quadrant did I have to select if this was missing over here? You would have looked at the percentage. Where are majority of your cells written? 86% of your cells are said to be in this quadrant. So either they will write a percentage, go by majority, or they will give you an arrow. And where is it? DP, double positive, matlab 10 be positive, TDT be positive. Of course, this is a case of BALL. Life becomes a lot simpler now. Basically, tha, you had to pick up that one CD markers ka table and you are sorted. Let's practice one more. What do I have? CD34 is mentioned over here. So blast, तो ठीक है, blast वाला marker. And 1333 किसका marker है? This is something to do with myeloid. 1333 was myeloid, and they have put an arrow in again the DP quadrant. Means C34 is positive, 1333 is positive. Blast भी है और myeloid blast है. What is your answer? Your answer is gonna be AML. Again, life is becoming easier. Let's move on. Let's move on to the next. What is your analysis? ये सबसे easy वाला है. This will give me a good question also. 59 and 55. As soon as I read these, I anyway know there is no other disease in pathology na that I can think of. CD55 and CD59. And what is the majority population? Majority population is DN, double negative quadrant. मतलब 90% cells तो यहाँ है. कुछ छोटा छोटा cells are over here, which I'll ignore. But 90% cells are here, double negative. CD55 and 59 missing has to be a case of PNH. But what is the take-home message from this? Which I'll take you back to anemias. Pehle to tell me the other names. CD55 might be written as DAF and CD59 might be written as MERL. So this is gonna be DAF and MERL. Ye wala pata hai point. Now, why did I see when you'll say, ma'am, if this is a case of PNH, all the cells should have been 55 and 59 negative. Why are 90% cells to hai, but why are these 10% cells coming positive? Because please note, PNH tends to show you a dual population. Go back to your anemia chapter. Majority of the cells are going to be 55, 59 negative, majority of the cells, but a tiny population of cells keeps the 55, 59. ऐसा क्यों हुआ? If it is a genetic problem, everyone should have been negative. Why are some cells positive? Is PNH a hereditary disorder from your parents, or is it an acquired condition? I hope you remember, in the anemias, this is the only acquired RBC defect. This is the only acquired RBC defect, means this is a problem that has not come hereditary. It has not come like hereditary spherocytosis, autosomal dominant recessive. ऐसा कुछ नहीं है. This is an acquired defect. It has not come from the parent. So if it is acquired, some of the cells will be affected, some of the cells will be normal. That is why they say PNH के लिए the gold standard is in front of you, flow cytometry, and you get a dual population. Sorted? Okay, let's move on to the next question. Again, when I read the markers, half my battle is won. 41 and 61. Anyway, I read 41 and 61, 4161. I know ये कुछ mega karyocyte and platelet वाला problem है. And majority of the cells are in the double negative quadrant. So negative, negative, 41 negative and 61 negative. मतलब GP2B3A is gone. And GP2B3A is gone is gonna be a case of Glanzmann thrombasthenia. Okay, someone asked me, Dr. Soumya, any class, after this you don't have to read flow cytometry from anywhere. This is the class. There is no other class that you have to refer to. You listen to this class from the beginning, all of your flow cytometry is sorted. Okay. Now we move on further. There are going to be sometimes they'll try to confuse you. Thoda trio wala questions aega. So what is all this that you have to study? So now again I'll enlarge this picture so that you guys can see it in a better way. And what are the things that you're seeing over here? Markers solves your story. You have CD5 written, so I can write it for you. There is CD5 written over here, and there is CD23 here. Then there is 20 and 10, and there is 5 and 200, which is written over here. So now tell me, sometimes you don't have to go through all. Even if you look at one or two graphs, na answer mil jayega. तो सबसे पहले तो ma'am, as soon as I saw 5 and 23 वाला profile, that table came to my mind. कौन सा वाला table? The one that we just now did, ये 523 वाला table that we did na right now, that should come to your mind. Where are all the cells coming? All are coming in the DP quadrant. All are coming in the DP quadrant. मतलब सब कुछ positive. 5 is also positive. 23 is also positive. यहाँ से 200 is also positive. When सब कुछ is positive, this has to be a case of CLL, SLL. So that is why I said na CD markers आता है तो flow cytometry is a cakewalk. सब कुछ positive means this is a case of CLL, SLL. Practice one more. Have a look at this. Same graph. The same graph is there. This is going to be 5. This is 23. This is 20 and 10. This is 5 and 200. But now the cells have changed. The position has changed. Where are they present? यहाँ पर only 5 is coming positive. See, it's present in this quadrant. Only 5 is positive. 23 is not positive. 23 negative आ रहा है. यहाँ पर भी if I look, only 5 is coming positive. 200 is not coming positive. So you will say, ma'am, all in all, I realized only one marker is positive, and that marker is CD5. Only one marker is positive. Rest everything is negative. This means it is a case of mantle cell lymphoma. Getting a little bit of a confidence, I hope, yes, because now I'm left with the last two, जो थोड़ा सा might be complicated, but we will crack it. It's nothing very difficult, right? अब इसमें क्या लिखा है? Firstly, as soon as I read 38, 138, तो I know कि अच्छा 38, 138 मतलब they are wanting to go towards plasma cells. इतना तो clear हो गया. तो बाकी options तो हटाओ. Plasma cells के बारे में they want to ask me. So either they are normal वाला plasma cells or they are cancer वाला plasma cells. बस दो ही options हैं. So when I say 38 and 138, over here 38 and 138 DP quadrant, both are positive. So 38 is positive, 138 is positive. But 38, 138 to every plasma cell, whether it is a normal plasma cell or is it a cancerous plasma cell, दोनों में आएगा. So now this didn't solve. I had to go to the next one. The next one is showing me 19 and 38. हाँ, this will tell me where are the cells present. यहाँ पर present है तो 38 तो positive है, but 19 has come out to be negative. He has forgotten his parents. He has forgotten his parents means he is a bad child. Bad child मतलब cancer वाला child. This means this is a case of neoplastic cells. This is a case of multiple myeloma. Sorted for everyone. Last question, I've always kept the celebrity for the end, and the celebrity is gonna be this, because this is a question which I receive over Telegram, Instagram after every few days. This is like a mandatory question which students have to ask na. So this comes in every grand test also, and same photo aata hai. So I'll first tell you how to interpret it, because that is something that most students don't read in the question ke karna kya. I know all of you have understood कि मैम अब तो इतनी बार वी हैव सीन दिस, वी नो दिस इज अ केस ऑफ हाइपर आईजीएम, बट कैसे बना हाइपर आईजीएम? सो प्लीज नोट, the first two graphs that have been given are of a normal person, any normal person like you and I. The second two graphs that have been given are for a patient. क्या क्या लिखा है? 19 is written and some site scatter is written. अरे site scatter से तो I'm not bothered. And examiner is nice ना, he has labeled red वाला cells B cells है, बस एग्जामिनर ने लेबल करके दिया रेड वाला सेल देखो, रेड वाला सेल ही कॉन्सेंट्रेट करो, रेड वाला सेल इज बी सेल. सो व्हेन आई गो टू द नेक्स्ट पार्ट, आई सी अच्छा 19 एंड यहाँ पे लिखा है 40. I'm writing it because it might not be visible. 19 and 40. And red wala cells are in the DP quadrant. So we realize 19 is positive, 40 is positive. Every normal B cell has that. Na? I told you, na, B cell mein double karo, 10s, 20s, 40s. So every B cell on this planet is gonna have 19 and 40 positive. Matlab ye normal B cell hai. So normal person tha bilkul. Now when I go to the patient, red wala cells focus karo. Where are the red wala cells coming? If this is 40 and this is 19, red wala cells are coming in this quadrant, means in a patient 19 to is coming but 40 is not coming. And what is that disease when 40 is going to be missing? 40 is going to be missing in a condition known as hyper IgM. So that is how you do the analysis. देखो, this is the easiest graph because examiner has labeled for you कि red वाले cell पे focus करना है. Red वाला cell is B cell, just focus on that. When you were getting 19 and 40 both, they were normal. Now you're not getting 40, matlab this is abnormal. This is the case of hyper IgM syndrome. Yes, everyone sorted. Someone in between was asking for the PDF. When have I not given the PDF? तो इसका भी तो मिल जाएगा. There is no doubt about it. But yeah, that comes to the end of this session. And as I said in the beginning, this session was only and more important for the INI-CET students. FMG does not get you so many questions. But yes, going forward, when I'll be meeting you on March 30th and from April 3rd to 6th, this is going to be a common session for all. Timings for all are gonna be 11 a.m. So don't keep running around thinking when and where is the class gonna happen, right? So that is there. Someone asked me a question. CD 154, yes, that is also there, Dr. Arun, perfectly. Dr. Simu, one is somatic hypermutation. Yes, somatic hypermutation, AICD. AICD is what you're probably asking me. Too much of detail that is. It's an enzyme that can be deficient sometimes in hyper IgM syndrome, right? Yes, these are all going to be YouTube sessions, and of course ये तो सारे TND वाला sessions है, ठीक है? When I go all the way to the end of April, I'm going to have 3 image based sessions, but that is towards the end because I believe closer to the exam you need a review of all the images, right? Abhi to if you read images you'll forget. Closer to the exam we'll flip through all the images once. Flow cytometry for NEET, yes Dr. Priya, it's important, of course. NEET and INI के लिए तो important है. FMG के लिए only the theory part is important, the graphs are not very very important. Okay, now thankfully, yes, we'll be meeting more often because I think it's been a while that we had classes, but yeah, these are the different things that are going on the platform for the time being. If you're planning for the NEET or the next wala batch, there is a batch that is starting on March 28th for that, and that is a special offer that is running. So this is the batch that I am talking about. This is the NEET or the next wala batch which is available, and this is starting, as I said, on March 28th. So this is going to be full 9 months wala batch that shall be going on. I will be taking path and micro under this. And by the way, this is another very important update I wanted to give you. I hope you all are aware about the NEET PG Vitals. NEET PG Vitals is nothing but the recorded, proper recording, not recorded classes, proper जैसे app recording होता है, that kind of a recording. And I have already finished the recordings for Path and Micro, and complete Path and Micro of the NEET PG Vitals has been uploaded on the Vitals. So if you go for this, you get access to the Vitals also. ठीक है? Will there be a micro class? Micro class, yes. Dr. Lekha, this series na April 3rd to 6th. So 3rd and 4th we have patho, and 5th and 6th we have micro. ठीक है? Next question, I have a query. What is a T regulatory cell? Yes, we can discuss that. Let me just bring up a blank slide over here. T regulatory cells are again important for INI-CET. Why? Because immunity इनका favorite chapter है ना. T regulatory cells is the only T cell. Normally, what do I say? T cells help in my immunity. This is the only T cell which is immunosuppressive. It has immunosuppressive action. So you will say T cell matlab ma'am, is it CD4 or CD8? It is a CD4 type of T cell, but it has something extra. It has CD25 and it is regulated by the FOXP3 gene. CD25 and FOXP3 gene, that is the T regulatory cell. Okay, everyone? Yes, Dr. Arun, perfectly correct. Is this okay? Done. CD4, CD25, FOXP3 gene, and this is the only T cell which suppresses our immunity. Rest all the B T cells, they increase or they fight for our immunity, right? ठीक है, PDF will be there. Give me 5 minutes to convert this PDF and to put it up on Telegram. So you'll get it from there. Not a worry. Um, Yes, the third to sixth wala are gonna be basically what I've decided for the third to sixth wala classes. I wanted to pick up the top 20 topics of path and top 20 of micro. And how have I decided? Aisa koi matlab I am not getting some paper leaked out from somewhere. I have decided the top 20 topics on the basis of the PYQs. So the previous year topics I've picked up in the last 5 sessions, and whichever is the most important in order and priority, I'm gonna discuss those in these sessions. Okay? That is the entire concept behind these classes, and they're going to be short. I'm not aiming for you're anyway doing targets on a daily basis. I'm anyway giving you already one hour ka study material every day, so I don't want to give you another 3 hours of lectures. We are going to keep it concise so that we can finish off the PYQ we can touch upon, right? Okay, perfect. So duration of those classes, as I said, as short. I'll try to wrap up within 1 hour. Okay, chalo, ठीक है. Thank you so much for joining in, and I hope this session was beneficial for all of you, because we know flow cytometry is one of their sought after topics. So thank you so much for listening out, and PDF I'll share with you within 5 minutes.